Welcome to the Microbiology and Immunology, V-1, Unit-3, Clinical Virology. 1. Multiplication process of viruses. Viruses are obligate intracellular parasites, they replicate only inside host cells. The general steps of viral replication include 1. Attachment, adsorption, virus attaches to specific receptors on the host cell. 2. Penetration, entry of the virus or its genetic material into the host cell. 3. Uncoating, viral capsid is removed, releasing the nucleic acid. 4. Replication, viral genome is replicated using host cell machinery. 5. Transcription and translation, viral mRNA is produced and translated into viral proteins. 6. Assembly, new viral particles are assembled from replicated genetic material and proteins. 7. Release, new virions are released via lysis, non-enveloped viruses, or budding, enveloped viruses. 2. Common and emerging viral diseases. Common viral diseases. Influenza caused by influenza virus. Hepatitis A, B, C, affects the liver. Herpes simplex virus, HSV, causes oral/genital sores. HIV/AIDS weakens the immune system. Dengue, mosquito-borne viral infection. Emerging viral diseases. COVID-19 caused by SARS-CoV-2. Zika virus transmitted by Aedes mosquitoes. Ebola virus causes hemorrhagic fever. Nipah virus, zoonotic virus, high mortality. Hantavirus spread by rodent droppings. 3. Basic laboratory procedures in viral diagnosis. Specimen collection, blood, throat swab, CSF, etc. Viral culture, growing virus in cell lines. Serological tests, ELISA, Western blot, detect antibodies/antigens. Molecular techniques, PCR, RT-PCR, detect viral RNA/DNA. Immunofluorescence, visualizing viral antigens using fluorescent dyes. 4. Cleaning and sterilizing glassware in virology lab. Cleaning. O. Rinse with distilled water after use. O. Use detergent and brush for thorough cleaning. O. Rinse several times with distilled water. Sterilization methods. O. Autoclaving 121 degrees Celsius, 15 Ci, 15-20 minutes. O. Dry heat 160-170 degrees C for 2-3 hours, e.g., hot air oven. O. Chemical sterilization, ethanol, formaldehyde for heat-sensitive items. 5. Gamesa staining for viral inclusion bodies. Purpose: to detect viral inclusion bodies in infected tissues or cells. Steps: 1. Prepare a smear on a clean glass slide. 2. Fix with methanol for 3-5 minutes. 3. Stain with Gamesa solution, diluted 1:10:1:20 with buffer, for 20-30 minutes. 4. Rinse with buffer/distilled water. 5. Air dry and examine under a microscope. Observation: inclusion bodies may appear purple or blue. Useful in detecting viruses like cytomegalovirus, CMV, herpes virus, etc. MCQs. 1. Which step of viral replication involves the virus entering the host cell? A. Attachment. B. Penetration. C. Uncoating. D. Assembly. Answer: B. 2. What type of virus causes AIDS? A. Herpes virus. B. Influenza virus. C. HIV. D. Hepatitis B. Answer: C. 3. Which of the following diseases is mosquito-borne? A. Hepatitis C. B. Dengue. C. Influenza. D. Herpes. Answer: B. 4. What is the purpose of uncoating in the viral cycle? A. Virus enters the host cell. B. Capsid is removed to release genome. C. New viruses are released. D. Proteins are synthesized. Answer: B. 5. Which diagnostic method detects viral RNA or DNA? A. ELISA. B. Immunofluorescence. C. PCR. D. Culture. Answer: C. 6. Which virus caused the COVID-19 pandemic? A. Zika virus. B. SARS-CoV-2. C. H1N1. D. Hepatitis A. Answer: B. 7. What is used to sterilize glassware in most virology labs? A. UV light. B. Boiling water. C. Autoclave. D. Cold water. Answer: C. 8. Which of the following is an emerging viral disease? A. Influenza. B. HIV. C. Hepatitis B. D. Nipah virus. Answer: D. 9. GMSA staining is used to detect A. Viral RNA. B. Antibodies. C. Inclusion bodies. D. Bacterial colonies. Answer: C. 10. What temperature is typically used in dry heat sterilization? A. 80 degrees Celsius. B. 100 degrees Celsius. C. 121 degrees Celsius. D. 160 degrees Celsius. Answer: D. 11. Which of the following viruses is transmitted via blood? A. Dengue. B. Influenza. C. Hepatitis B. D. Zika. Answer: C. 12. Which staining method is commonly used for detecting cytomegalovirus inclusion bodies? A. Gram stain. B. Acid-fast stain. C. GMSA stain. D. India ink. Answer: C. 13. During which step of the viral cycle does the virus attach to the host cell surface? A. Penetration. B. Release. C. Assembly. D. Attachment. Answer: D. 14. Which of the following is not a method for viral diagnosis? A. PCR. B. Viral culture. C. Gram staining. D. ELISA. Answer: C. 15. The term budding refers to A. Viral entry into host cell. B. Release of enveloped viruses. C. Lysis of host cell. D. Mutation of viruses. Answer: B. 16. Which virus is associated with hemorrhagic fever? A. Hepatitis A. B. Ebola virus. C. HSV. D. CMV. Answer: B. 17. Which of the following is a serological test? A. ELISA. B. PCR. C. GMSA stain. D. Culture. Answer: A. 18. What color do viral inclusion bodies appear with GMSA stain? A. Green. B. Yellow. C. Purple/blue. D. Red. Answer: C. 19. Which virus is transmitted by rodents? A. Influenza. B. Ebola. C. Hantavirus. D. Zika. Answer: C. 20. In viral replication, what happens during the assembly stage? A. DNA replication. B. Virus enters host cell. C. New viral particles are formed. D. RNA transcription. Answer: C. Like, share and subscribe.